My name is Matthew Altman. I'm a physician and a research scientist in the Benaroya Research Institute, Center for Systems Immunology. I'm part of the NIAID Childhood Asthma in Urban Settings, or CAUSE network. The longstanding focus of this network has been to understand how the environment and host molecular factors interact with the body's immune system to both cause asthma and aggravated symptoms in urban and underprivileged children. Fundamentally, the network seeks to both prevent and better treat asthma, especially in these populations. I'm honored to share our manuscript published in the Journal of the American Medical Association Pediatrics, entitled Inflammatory Pathways in Residual Asthma Exacerbations Among Mepolizumab-Treated Urban Children, a secondary analysis of a randomized clinical trial. It's a big title. Fundamentally, what we sought to do was to understand precisely why some children respond, and perhaps more importantly, why some fail to respond to an important biologic therapy for asthma. We did this by studying the nasal airway inflammatory patterns during respiratory illnesses and asthma exacerbations in children enrolled in a randomized controlled trial utilizing the drug mepolizumab. We linked transcriptomic profiling, which is collected by sequencing all expressed genes in nasal samples, to respiratory illness outcomes and to lung functions. We were able to pinpoint the inflammatory patterns underlying the residual asthma exacerbations in children with a poor or incomplete response to the therapy. In particular, highlighting alternative, so-called non-T2 immune pathways. These pathways link, in many cases, to inflammatory, remodeling, and mucus secretion responses of the airway epithelium, and in other cases to infiltrating immune cell pathways, notably those related to macrophages. These pathways play an outsized role in asthma exacerbations after aspects of T2 and eosinophilic inflammation have been blunted by mepolizumab. Importantly, these pathways indicate treatable targets that can and are being pursued towards novel and more personalized treatment strategies. Specifically, treatments that could better address the large gaps that exist in mitigating the urban pediatric asthma epidemic, which afflicts a highly vulnerable patient population. Furthermore, our results highlight the importance of incorporating longitudinal, systems-level molecular data capture into a clinical trial to provide in-depth assessment of mechanisms that could lead to predicting response or failure to a molecularly targeted therapeutic at the level of the individual. Based on this and other ongoing works by our network, we are advancing the study of additional therapeutic strategies in this population with the goal of accessible, effective, and personalized treatment strategies to mitigate the morbidity and mortality of asthma in urban children. Thank you for your interest.